Alright, biology nerds. It's time for Unit 2. Found this nice biological specimen here. They call that a unicorn. Hopefully it's not a premonition of your ability to get a 5 on this test. Alright, I'll try to keep this concise. Unit 2 is all about the cell and the structures in the cell. Hopefully this is all like, oh yeah, I know this. No problem, skip ahead, please. Nucleus, control center of the cell, contains the DNA and the nucleolus, a little structure here that makes RNA, ribosomes, and you can see it's perforated with pores. This is going to allow the transcripts to leave the mRNA when it leaves. Anyway, we have the endoplasmic reticulum, this highly folded surface next to it. This particular image is only rough endoplasmic reticulum. I don't really see smooth. Oh, there it is. Remember, rough endoplasmic reticulum is where proteins are going to be made in the surface or into the membrane here, and you're going to have all these proteins made. I'll get back to that in a minute. Smooth endoplasmic reticulum, still highly folded, no ribosomes, has many function, lipid production, calcium storage, testosterone, estrogen production, etc. But this protein inside the rough endoplasmic reticulum is going to travel to the Golgi apparatus, usually in a vesicle. It'll fuse. Golgi apparatus, the post office of the cells, where the protein will be modified, activated, and whatever needs to happen. It'll form a vesicle as it leaves, and then if it is meant for some other organelle or some sort of internal use, it'll go to that organelle, that location, on these little fibers. It's kind of like zip lines. And then if it is meant for release, it'll fuse with the plasma membrane and be released outside the cell. This is also the Golgi apparatus, and this vesicle can also add components to the plasma membrane. So it helps replenish the plasma membrane because this vesicle is a phospholipid, just like the membrane. The lysosome, this guy here, remember, it has those hydrolytic enzymes, hydrolysis, which is breaking down those macromolecules. That hydrolytic reaction, it's got a very low pH, so if it bursts open, those enzymes are inactive inside this cytoplasm, cytosol, and don't work, so it doesn't eat up the cell. What else am I forgetting? I feel like I'm forgetting one. The ribosome makes the proteins. Yeah, I took a moment to think, and the only ones that are left then will be the mitochondria and the chloroplast. All right, mitochondria and chloroplast. Mitochondria, as hopefully you well know, is the site of energy production, cellular respiration. And the structure you should know, here you've got the outer membrane, and then you have the inner membrane space. Then you have the inner membrane, which is called the cristae, these little gray with light tan folds. And then the matrix is the inside of the cristae, and it's this mechanism, this movement of H plus through these, between the matrix and intermembrane space, that drives the production of ATP, right, using ATP synthase. So that's the mitochondria. Special features of the mitochondria and the chloroplast: they have the double plasma, they have the double membrane, and they have their own internal DNA. All right, the chloroplast and its structure, you got the fluid, which is the stroma, you got the inner and the outer membrane, and then you have the thylakoid, which is the one, and then the stack of these is called the granum. That's the structures in the cell. Quick review. Limiting the cell size, right? The cells are pretty small, and it's the surface area to volume ratio. You want to maximize your surface area and volume by, you can do calculations. I tried to do some formulas here, and it wasn't formatting right. I'm sure you can just look them up. But you want this number to be the higher number, right? Surface area to volume, surface area divided by volume gives you your ratio. So here the 6, and there we've got this stack that they have, and it's kind of broken down, but it's the same surface area each. And then this one, which has a large surface area and a larger volume, but still The ratio is 1.2, which is much less. And the reason you want to maximize that surface area to volume ratio is for transportation. Right? It's easier for things to diffuse through and into the cell and get to where they're going if you have a higher surface area, less volume, and that's why you have things that are highly folded, like this endoplasmic reticulum. That's my dog barking. And you have even the mitochondria surfaces that are folded. So hopefully that is a piece of cake. All right, plasma membrane. All right, it's a phospholipid bilayer, and it's surrounding every cell that exists. And you have got the hydrophilic heads pointing out towards the water, and the hydrophobic core internal, and then mixed in the plasma membrane, you've got these channels, and you've got different kind of surface receptor proteins and molecules that are inside and outside. The hydrophilic head will attract to water and things, but will repel hydrophobic stuff. The only thing that really can get through is small nonpolar molecules, nitrogen, oxygen. They can freely diffuse a little, very slowly, but even water's got its own little pore. And so it's beautiful because things that kind of get through, if the hydrophilic things hit that hydrophobic tail, that lipid part, and they get expelled out. I threw this diagram on here to kind of show you, different transportation mechanisms. I think it looks like maybe I pulled up I didn't look at this well enough. But you have transport proteins, right? You have these ion-gated channels. You've got just tunnels. They can open or close, and they're very specific to the molecule, and they'll let things in or out. You have things that can, signaling receptors up there that attach. We got the G protein receptors. We can also have signal molecules attach, and they can also be ingested in. But there are many different ways for molecules to get in. The channel is a good way, main way that many do. Speaking of transportation, we have passive and active. Passive is high to low, no energy required, diffusion, osmosis. So in this example, with the ions, I have more outside than inside, so it will be passive going into the cell. These four, so balance out, you end up with 1.5. Wrong way. Active is always low to high. ATB is required for this to happen. So in this example, active transportation would be taking this ion and pumping it out. It would require some sort of ATP. Think of the sodium-potassium pump, where you have the molecules attached on either side of the membrane. Then ATP comes along, and that ATP changes the shape of this protein, which then releases the ion either into or out of the cell. And then the cleaving of the phosphate changes the protein shape again, and either one of them is released and one of them is not. Again, it goes back to shape. These proteins here are very sensitive to shape and are going to be very specific to the molecules that they are transporting. Oh, that's it. Look at that. 8.5 minute review. I did it quickly, but it is review. So good luck and enjoy your day.